DNA. Get your genes on, get your genes on, yeah, yeah. DNA and RNA. Get your genes on, get your genes on.
Hello, my name is Rosalind Franklin, and welcome to my science lab. You're most likely wondering who I am. I'm best known for making the very first X-ray diffraction of DNA. I was just about to check on DNA and RNA right now. Here, have a look.
Hi, I'm deoxyribonucleic acid, known as DNA, and this is my friend ribonucleic acid, or RNA.
Oh, wait, we're late to class. Let's tell the viewers a little bit about ourselves.
Oh, that's a great idea. My name is DNA, which is short for deoxyribonucleic acid. I'm made up of nucleotides joined in long strands or chains by covalent bonds. I contain deoxyribose, and I'm double-stranded. My nucleotide pairs are AT and GC, which stand for acetine, thymine, guanine, and cytosine. I have one type, and I store, copy, and transmit information.
I'm RNA, also known as ribonucleic acid. I consist of long chains of nucleotides. I contain ribose, and I'm single-stranded. My nucleotide pairs are AU and GC. I have three types: messenger, transfer, and ribosomal. My function is that I do protein assembly synthesis.
Oh, here we are at the history classroom. Come on in.
Hello, my name is Miss Lavonsky Monroe. Welcome to my class.
Hey, can I see your notebook or your notepad?
Yeah, sure.
Thanks. Hey, this is just like the central dogma for molecular biology.
Oh yeah, it is. Doesn't it state that information is transferred from DNA to RNA to protein?
Yeah, that's right.
Please pay attention. Let's go back to what we were talking about before.
Oh, sorry.
Well, we were talking about the contributing sciences, and the first one was Gregor Mendel. He discovered that the characteristics of pea plants are passed along in a predictable way. His discovery begins the science of genetics. Next scientist is Walter Sutton. He showed that chromosomes carried the cell's unit of inheritance. Thomas Hunt Morgan demonstrates that genes are arranged in linear fashion on the chromosomes of the fruit fly. Erwin Chargaff analyzed the base composition of DNA in cells. He discovered that the amounts of adenine and thymine are almost always equal, as are the amounts of guanine and cytosine. Alfred Hershey and Martha Chase confirmed that the genetic material of viruses is DNA, not protein. James Watson and Francis Crick published their model of the DNA double helix. The model was made possible by Franklin's work. This is an example of the model of DNA that Francis Crick and James Watson made.
Miss Lavonsky Monroe,
Yes.
what's the Human Genome Project again?
Does anybody want to answer that?
Oh, I do. The Human Genome Project is a 13-year international effort with the main goals of sequencing all 3 billion base pairs of human DNA and identifying all human genes.
Thank you. Oh, it's time to go to science class. Come on.
Have a nice day, young children.
Welcome to my class. Please stand up and state your name.
RNA, I'll tell you about me, RNA. I come in 3 different forms: messenger, transfer, and ribosomal too. You'll learn more about me as I spend more time with you.
DNA, you know about me, DNA. I store and copy information, and I transmit info too, and all that happens to living beings just like you.
Nice to meet you.
Today will we be learning about humans and how we ruin their lives?
Of course.
Ready.
Today we will be learning about humans and how we ruin their lives. An example will be mutations. There are two types I'm going to be talking to you about today. The first one is gene mutations. That is when it produces one change in a gene, and there's also chromosomal mutations, when it creates changes in whole chromosomes. Another way that we ruin their lives is by disorders. One disorder would be sickle cell anemia. Is when cells become the shape of sickles and it blocks your blood passages. Another one is cystic fibrosis, when your cells will not let the carbon dioxide secrete from your cell wall. And then the last one I'm going to talk to you about is Huntington's disease. Is when when a chromosome inside of your brain has a defect and it creates abnormalities. I want to go sleep now, so...
Okay, class, it's time to study for your vocabulary test.
All right, transformation.
A process in which one strain of bacteria is changed by a gene from another strain of bacteria.
All right, yeah.
Bacteriophage.
A kind of virus that infects bacteria.
Yep.
Base pairing.
A principle that bonds when in DNA can form only between adenine and thymine and between guanine and cytosine. Okay, replication.
A process of copying DNA prior to cell division.
Right, okay.
Telomere.
Repetitive DNA at the end of eukaryotic chromosomes.
Perfect.
Okay.
DNA polymerase.
Principal enzyme involved in DNA replication.
Perfect.
Okay. Ribonucleic acid.
Oh, wait. All right, a single-stranded nucleic acid that contains the sugar ribose. That's you.
Yeah. Okay.
Transcription.
Synthesis of an RNA molecule from a DNA template. Okay. RNA polymerase.
I got this one.
This is a tough one.
Enzyme that links together the growing chain of RNA nucleotides during transcriptions?
You forgot using a DNA strand. That was close.
Ah.
Okay.
Promoter.
A specific region of a gene where RNA
Polymerase?
yeah, can bind and begin transcription. Yeah. Oh, polypeptide.
Long chains of amino acids that make proteins.
Right.
And genetic code?
This one's kind of tough. A collection of
Codons.
Codons, right. Oh my goodness, you're so smart. Of messenger RNA, each of which directs the incorporation of a particular something acid.
Amino.
Yeah, amino acid in a protein during protein synthesis. Cool. Okay. Here, your next word is translation.
That's a process by which the sequence of bases of an messenger RNA, yeah, is converted into the sequence of amino acids of a protein.
Good job.
And mutation.
A change in the genetic material of a cell. It's of a cell.
Yeah. Perfect. All right. I think we're ready.
We're ready.
Sorry, class. The quiz actually didn't print out, so we'll actually have it tomorrow, but at least you learned something.
Yay!
Hello. My name is Miss Hululululu. Today we're going to be learning about pedigree, which is the record of always transferring from parents to offspring.
Hi, Miss Hulu. I have a message for DNA.
Oh, why don't you tell the class who you are?
Sure. Hi, my name is messenger RNA. I'm a type of RNA that carries copies of instructions for the assembly of amino acids into proteins from DNA to the rest of the cell. I also have a little sister named rRNA. Her full name is ribosomal RNA. She is a type of RNA that combines with proteins to make ribosomes. I have to make many more messages. I'll see you later, Miss Hulu.
Okay, bye, see you later. Class, please welcome our new transfer student.
Hi, I'm transfer RNA. I'm the type of RNA that carries each amino acid to a ribosome during protein synthesis.
Oh. Oh, look, on my paper laptop. I just found out that Mrs. Ribosomal DNA and her husband, Protein, who came together, have a ribosome now. Isn't that wonderful? Yay!
Hey, we should go over those vocabulary words we learned in homeroom.
Oh yeah, that's a great idea.
Okay, karyotype.
Karyotype is the characterization of the chromosomal complement of an individual or a species, including number, form, and size of the chromosomes.
Yeah, that's correct. Okay, my turn.
Okay, intron.
A segment of a gene situated between exons that is removed before translation of messenger RNA and does not function in coding for protein synthesis.
All right, good job.
Okay,
Forward.
codon.
Okay, codon is a sequence of 3 adjacent nucleotides constituting the genetic code that determines the insertion of a specific amino acid in a polypeptide chain during protein synthesis, or a signal to stop protein synthesis.
Yep, that's correct.
Great. Okay, your word is autosomal.
A chromosome that is not a sex chromosome.
Good.
And then extron.
Extrons are markers on a DNA strand that help us realize the different parts of the cell, right?
Yeah.
Okay.
Oh, well, class is done.
Bye!
Scene 2. Take 1. Okay. Please rise and say your name.
That was pretty hard.
Scene 2. Take 2. Welcome to my class.
Joli. Joli.
Welcome.
We need it. Okay, Joli.
Scene 5. Part 2. Take 3. Action.
Hey, we should go over those vocabulary words we were working on in homeroom.
Yeah, that's a great idea.
Okay, karyotype.
The characterization of the chromosome.
Some. What are you doing?
No, she hit something. Okay, do
I don't
it
know what
again.
happened.
Do it again.
stuff going on.
Okay, do it one more time.
Deshaun's having so much fun in the background.
Yeah.
Oh yeah, that's
Okay,